So this is Mr. Mann here for the honors biology class. So the first question we were going to answer before we started, as I get back to the beginning, is kind of reviewing for summer work what two parts make up a virus. And so that's one of the things we're going to be able to answer as we go through. They're just kind of helping you guys with summer work. We're going to start with viruses. So we should be able to identify the structures and shapes of viruses and describe different types of viral infections. And then we can even talk about classes as well within this identifying structures and shapes. And so one thing I'm going to tell you is with scientists, we're not going to really know or really need to know unless told otherwise. This just kind of gives us a little intro or a little background into viruses specifically. So in 1892, Dmitri found there was an infectious agent in tobacco plants, and it was smaller than a bacterium. And so within this, it was kind of created some issues within these farmers. And so it was something that we could treat bacteria, we could treat fungi, but we didn't really have anything for these viruses. And so in 1998, Martinez proposed that there was a small particle within the extracts that caused infections. And they named them viruses from the Latin word poison. And, anything from this last year and a half can really show you what that looks like within these viruses being poisoned. Now, we don't speak Latin anymore, but at that point in time, it was a great word to be able to describe what viruses are. And it was very relatable. And so viruses, they have a very basic structure. The biggest thing is a single viral particle is known as a virion. And so that's just one, COVID-19 particle is a virion, while more than one would be the COVID-19 virus. And so a virus is composed of two major things. The first one is genetic material, whether it be DNA or RNA, depending on the class or group. And the other one is a capsid, which is a protein shell that covers. So as we can see here with a capsid that contains this DNA here, while the DNA is found here, or it could be RNA. And then other times, depending on the virus, a lipid envelope, as you can see here, looks like kind of like a cell membrane previously. And it's a protective outer coat. And remember, lipids are fats. So again, it has to contain genetic material, DNA or RNA, and a capsid, protein shell. But then sometimes it can be, we can have a lipid envelope as a protective outer covering. So viruses can replicate in multiple ways in a host of a cell. And the first one we're going to talk about is the lysogenic infection. And it does no immediate harm to the cells. And so within this, the viral DNA is called a prophage when it combines, scratch that. First thing that's going to happen is we have a virus. Okay? It's going to inject its DNA into a host cell. So for instance, a host cell would be human cell, dog cell, whatever. And that cell or that viral DNA is going to intertwine its DNA into the host cell's DNA. And so every time that the cell replicates, it's going to replicate the viral DNA. And so as it replicates, the prophage, or the viral DNA after it's being injected to the host cell, replicates along with the host cell's DNA. After many cells for division to produce a colony of bacteria infected with this prophage, then the viral DNA is called a prophage when it combines with the host cell's DNA, which we've already talked about. And then there's just a vicious cycle. So after we go reproduce, we're going to be able to cause harm to this organism, because we're going to be making proteins and things from this viral DNA. Now we have a lytic infection cause the host cells to burst. And so first off, with the host bacterium, the bacteriophage is going to infect its DNA into the host cell. The bacteriophage attaches and injects its DNA, and this is all through injection here into the host bacterium. From there, the viral DNA forms a circle. Then the viral DNA directs the host cell to produce new viral parts to assemble new bacteriophages. And then the host bacterium breaks apart or lyses, lytic cycle, lyses. Then the bacteriophages are able to infect new hosts. So basically we're sacrificing one bacteriophage to making of the many. to infect these cells. And these are what's going to cause death a lot of the times within populations. So, kind of going on with that, the lytic cycle is known as virulent. So virulent viruses destroy the cells that they infect. And the viruses who replicate through the lysogenic cycle are known as temperate viruses. And the viral DNA is integrated into the host cell's chromosome known as the provirus or prophage. Maybe all it takes is the viral DNA, the host cell DNA intertwine, prophage. Now, each virus has different characteristics. And so we're going to classify them, whether they're DNA or RNA, or whether it's single-stranded and double-stranded. Go back here just for a second. And so within this, okay, we have 7 different classes. And we call this the Baltimore classification system. So 7 classes. We'll kind of go through those very, very quick and simplistic. Now, the biggest thing I need you to know is the class and whether it's DNA, RNA, single-stranded, double-stranded, positive, or negative. Now, positive means that the RNA or DNA is the exact copy, while negative means it's a complement strand. So complement would be like A's and T's, C's and G's. The T and the G would be complements of the A and T, or the A and C. So here's another picture of the same thing we're looking at. The biggest thing is getting to mRNA because mRNA makes protein, and proteins is life for cells. If we don't have proteins, we don't have anything. And so the whole goal is to get to mRNA so we can make those proteins for these viruses to survive and do what they're supposed to do. So our first class are double-stranded DNA virus. And it's going to host replication machine. It's going to use the host replication machinery, and some examples are the adenoviruses and the herpes viruses, or the herpes simplex. And so I'll show you what I'm looking at to try to help you. This is what I want you to know. I'm going to make this bigger screen for us. Right here. I need you to be able to fill in the groups and the characteristics. So for group 1 or class 1, the characteristic is double-stranded DNA. So going back. So class 2 virus is a positive single-stranded DNA. And this is an example is the parvovirus. And these intermediates are great, but I'm not going to hold you accountable to those. The biggest thing is knowing class 2 positive single-stranded DNA. Now, class 3. Class 3 viruses are double-stranded RNA viruses, or called arioviruses. So class 1, double-stranded DNA. Class 2, single-strand positive DNA. Class 3, double-stranded RNA. Now, class 4 are positive single-stranded RNA viruses, and one of the big ones that we all know is the coronavirus, or COVID-19, is a class 4 virus. So is the common cold. In class 4 viruses, again, it's positive single-strand RNA virus. Class 5 are negative single-stranded RNA viruses. So again, class 3, double-strand RNA virus. Class 4, positive single-stranded RNA virus. Class 5, negative single-stranded viruses. And rabies is a great example of class 5. Class 6, this is where it gets interesting. The biggest thing I need you to know with class 6 is this, is you can read the words within these retroviruses on the positive single-stranded RNA virus, which go through a double-stranded DNA intermediate, or you could say this. It's a positive single-stranded RNA virus with reverse transcriptase. Okay, and transcriptase is an enzyme, so make sure that you know that transcriptase is an enzyme. But all it's saying is going from RNA to DNA. It's going through reverse transcription. Class 7 viruses are double-stranded DNA, which will undergo a single-stranded RNA intermediate, or in other words, it is a double-stranded DNA virus using reverse transcriptase. And so that's all you need to know for class 7 as well. So then viruses cause a variety of infectious disease, common cold, influenza, SARS, HIV, all that stuff. And so what I'm going to do is I'm going to open up the whiteboard here real quick, and I'm going to show you all some stuff on some comparing and contrasting bacteria and viruses. It says my handwriting's terrible. I'm going to just do it this way. Bacteria. All right. Well, it looks like I'm going to have to write it. All right. So, bacteria. They are made up of cells. Viruses are not. Both viruses and bacteria are made of a protein. So this is my both. And they also contain genetic material. And this is where it gets a little crazy. Bacteria, inside where they store their genetic material, is going to be DNA. While on viruses, can be DNA or RNA. Bacteria can reproduce. And remember, this is a review from the activity we did in class the last couple days. And then, while viruses need a host cell to reproduce. Bacteria also uses energy. Or what some said, carbon. While viruses, again, use a host to do all their functions. And so that's kind of where we left off. We could even say instead of genetics, we could use nucleic acids. But this will be a question to where, you might have to give me 2 similarities and 3 differences. And from there, you guys will be able to kind of figure that part out just from looking at this. So again, bacteria has cells, viruses don't. Bacteria uses DNA, while viruses use DNA or RNA, depending on the virus. They both contain genetic material. They both contain protein, proteins in the capsid of the viruses, while proteins in everything else with the bacteria. Bacteria reproduce while viruses use the host to reproduce. Bacteria uses energy, expends energy, while viruses, again, use the cells with the bacteria or any host cells energy to be able to reproduce or do anything like to that nature. I hope this helps you guys. If you guys need anything, please let me know. And I will see you guys on Wednesday for our quiz for my Monday, Wednesday group, and for my Tuesday, Thursday group, which is my 2nd, 5th, and 7th period classes. You guys take a quiz on Thursday. Have a great day.